So we'll begin now with our second phase, second period of presentations with Dr. Gargano, who will present the grade for Moderna COVID-19 vaccine. Please, Dr. Gargano, go forward.
Good afternoon. Next slide, please. The policy question under consideration is: should vaccination with Moderna COVID-19 vaccine be recommended for persons 18 years of age and older under an emergency use authorization? Next. The components of the PICO question are shown on this slide. The population under consideration is persons 18 years of age and older. The intervention is 2 doses of the Moderna COVID-19 vaccine, 28 days apart. The comparison is no COVID-19 vaccine, and the work group identified the 7 outcomes listed here as the most important for the policy question. Next slide. The potential benefits outlined in red here included prevention of symptomatic COVID-19 and hospitalization due to COVID-19, which were both considered critical outcomes, and all-cause death, SARS-CoV-2 seroconversion, and asymptomatic infection identified as important. Next slide. The 2 harms identified were serious adverse events, a critical outcome, and reactogenicity grade 3 or 4, an important outcome. Next. Of note, in the case of the current COVID-19 phase 3 vaccine trials, hospitalization due to COVID-19 and deaths are less common, and the trials may not be designed or powered to evaluate differences between treatment groups. We do not necessarily expect to have robust direct evidence for these outcomes at this point, and to some degree can infer that decrease in symptomatic COVID-19 would also translate into decreases in hospitalizations and deaths, as has been observed for other vaccines. Next. For the outcome of asymptomatic infection assessed using serial PCR testing, data from 1 time point were assessed, and these were included in the evidence profile as preliminary evidence. Next. And finally, for the outcome of seroconversion, no data are currently available, so this could not be included in the evidence profile I'm going to present today. Data on seroconversion will eventually be available in an ongoing phase 3 trial. Next. We conducted a systematic review to identify evidence related to the policy question. We identified published articles using the databases and search terms listed here to identify relevant published literature. We sought out additional resources, including obtaining unpublished data from vaccine manufacturers. Next. Briefly, over 2,800 records were identified through database searching, and records for 2 studies were obtained directly from the trial sponsor. Ultimately, 4 studies were included in the evidence synthesis. Additional information on the identified studies was included from Food and Drug Administration materials prepared for the Vaccines and Related Biological Products Advisory Committee. Next slide. Grade evidence type assesses the certainty of the estimates from the available data. The highest level of certainty is type 1, which means we are very confident the true effect lies close to that of the estimate, down to type 4, indicating very low certainty, meaning we have little confidence in the effect estimate. The evidence type is not measuring the quality of individual studies, but how much certainty we have in the quantitative estimates of effects across each outcome. Next. Initial evidence type is determined by the study design. A body of evidence from randomized controlled trials starts with an evidence type of 1, indicating high certainty. A body of evidence from observational studies starts with an evidence type of 3, indicating low certainty. The evidence type can be downgraded due to risk of bias, inconsistency, indirectness, or imprecision. Other considerations could downgrade or upgrade the evidence type. Next. Okay, now we will review the evidence for the benefits. Next slide. For the critical outcome of symptomatic, laboratory-confirmed COVID-19, 1 study provided data. This was the Moderna phase 3 randomized controlled trial. Data were obtained directly from the sponsor and from FDA briefing documents. The primary data cutoff date was November 25th, 2020, but some analyses were only available from the planned interim analysis conducted using the November 11th cutoff date. Next. A total of 30,351 adults were randomized 1-to-1 to receive either the vaccine or a saline placebo. Analyses were performed using different data sets. The full analysis set included all randomized participants who received at least one dose, analyzed according to the group to which they were randomized. The modified intention to treat set excluded persons with immunologic or virologic evidence of prior SARS-CoV-2 infection at day 1 before the first dose, and the protocol set included persons from the modified intention to treat set who received both planned doses according to schedule and had no major protocol deviations. The sponsor used this per protocol set for primary efficacy analyses. Next. Using the per protocol population, for all persons aged at least 18 years, there were 11 cases among 14,134 persons in the vaccine arm and 185 cases among 14,073 persons in the placebo arm, which resulted in a vaccine efficacy estimate of 94.1% and a 95% confidence interval of 89.3% to 96.8%. This is the outcome that was used for grade. Next. Vaccine efficacy was also at least 86% in various subgroups, including those aged 65 and older, 75 and older, and those at risk due to presence of a comorbidity, including severe obesity. Next slide. This slide compares the primary outcome with some secondary outcomes, which are highlighted. Efficacy remained high when persons with evidence of prior infection were included and when a broader list of symptoms was used to define COVID-19. Next. I also want to show a few post hoc interim analyses. In the modified intention to treat set, in an analysis that was restricted to persons who received only one dose of vaccine or placebo and had no evidence of prior SARS-CoV-2 infection, there were 7 cases reported among 996 persons who received one dose of the vaccine and 39 cases among 1,079 persons who received one dose of placebo, for a one-dose vaccine efficacy estimate of 80.2% and a 95% confidence interval of 55.2% to 92.5%. Next. In the full analysis set, which did not exclude people with evidence of prior infection and included everyone who received at least one dose, the efficacy point estimate was 87.9% using all available follow-up post-dose 1, but we do recognize that the vast majority did receive the second dose. Censoring this analysis at dose 2, the one-dose efficacy estimate using the full analysis set was about 28 days, with about 28 days of follow-up, was 69.5%. Next. Here is the grade evidence table for the outcome symptomatic laboratory confirmed COVID-19. Because the data were from a randomized controlled trial, the evidence type started at 1. Regarding risk of bias, there was some concern related to blinding. Participants and study staff were blinded to assignments, but they may have inferred receipt of vaccine or placebo based on reactogenicity. This was deemed unlikely to overestimate the efficacy results, so therefore we considered the risk of bias not serious for this and subsequent outcomes. Some concern for indirectness to the outcome was noted due to the short duration of observation in the available body of evidence. The vaccine efficacy observed at a median 2-month follow-up may differ from the efficacy observed with ongoing follow-up. However, in consideration of the strength of association and precision observed for this outcome in particular, it is unlikely that the efficacy estimate for symptomatic COVID-19 would change substantially enough to fall below the FDA-defined efficacy threshold for licensure under an emergency use authorization, that is, to less than 50% efficacy. Next. Oh, wait. Go back, please. We also acknowledge some concern for indirectness to population because of exclusions from the clinical trial. We judge this to be not serious, in part because all available subgroup evaluations were so consistently really quite high. The relative risk of 0.06 with a 95% confidence interval of 0.03 to 0.11 indicated a strong and precise estimate. There were no other serious concerns affecting the certainty assessment. We assess the level of certainty as high or type 1 for this critical outcome. Next. The second outcome for consideration was hospitalization due to COVID-19. The protocol indicated a definition of severe COVID-19 with criteria shown on this slide, but this did not require hospitalization. Additional data on severe cases, including hospitalizations, was obtained from FDA documents. Of note, one severe case leading to hospitalization in a vaccine recipient was included in our analysis that was adjudicated after the November 25th data cutoff date. The sponsor indicated that hospitalization due to COVID-19 was not specifically ascertained for all cases, and it is possible that hospitalizations for COVID-19 in cases that did not meet the FDA's definition of severe COVID occurred and were not captured. Next slide. This slide shows analyses of both severe COVID-19 using the protocol definition and hospitalization for COVID-19. About 2/3 of persons meeting the severe COVID-19 definition were not hospitalized. For severe COVID-19, there was one severe case in the vaccinated group and 30 in the placebo group. The efficacy estimate was 97%, with a 95% confidence interval of 76% to 100%. For hospitalization due to COVID-19, there was one case in the vaccine group and 9 in the placebo group. The efficacy estimate was 89%, with a 95% confidence interval of 13% to 99%. This wider confidence interval reflects the smaller number of events, but it is still statistically significant. The analysis of hospitalizations due to severe COVID-19 outlined in red is the one used for grade, and it aligns with the PICO specified outcome. Next. This slide shows the grade evidence table for hospitalization due to COVID-19. The initial evidence type of 1 was downgraded 1 point due to serious concern over indirectness of outcome because of use of a subset of cases defined as severe using a set of protocol criteria, which may miss some hospitalized cases and the short duration of follow-up. COVID-19 leading to hospitalization measured in such a short time frame is an indirect measure. Some hospitalizations may not have occurred yet for some cases included in the analysis, and we would ideally be able to observe efficacy for preventing COVID-19 hospitalizations over a longer follow-up period. The final certainty estimate for the outcome of hospitalization due to COVID-19 is type 2. Next slide. Okay, the next outcome of interest was all-cause death, which the sponsor regarded as descriptive only. All-cause death was not an efficacy endpoint in the trial protocol, although death due to COVID-19 was. Next. There were few deaths among trial participants, including 6 among vaccinated persons and 7 among placebo recipients. No person-time analysis of deaths was provided for all-cause death, and the denominators here are based on the safety population of persons who received at least one dose. The available data indicates a relative risk of 0.86 with a 95% confidence interval of 0.29 to 2.55. Of note, there was one death attributed to COVID-19 during the available follow-up, and it was in a placebo recipient. Next slide. The grade evidence table for all-cause death is shown here. There was no serious risk of bias identified and no serious concern of inconsistency. There was serious concern for indirectness due to the short duration of follow-up. Deaths due to COVID-19 may not have had time to occur during the follow-up period. There was very serious concern of imprecision. The relative risk of 0.86 favored vaccination, but the very wide 95% confidence interval did not rule out harms. The certainty estimate was type 4. Next slide. The phase 3 trial provided some preliminary data regarding the outcome asymptomatic SARS-CoV-2 infection. The work group intended this outcome to be assessed longitudinally through serial PCR testing or other repeated laboratory assessment. As part of the trial protocol, a nasopharyngeal swab was collected for PCR testing for SARS-CoV-2 DNA at each vaccination visit. In the per-protocol analysis, these were used along with serology data to exclude persons with evidence of prior SARS-CoV-2 infection from the primary efficacy analyses. The sponsor used data on results of the dose 1 and dose 2 PCR tests, along with symptom data collected between doses 1 and 2, to construct a measure of asymptomatic infection on the day of the second vaccine dose. Using the data this way was not a predefined protocol objective. Next slide. Here are the data on the outcome of asymptomatic SARS-CoV-2 infection at dose 2, defined as a PCR positive nasal swab at dose 2 and no symptoms of COVID-19 during the interval between doses 1 and 2. In the vaccine group, there were 14 asymptomatic infections, and in the placebo group, there were 38 asymptomatic infections for a relative risk of 0.37 and a 95% confidence interval of 0.20 to 0.68, providing preliminary evidence that the vaccine may prevent asymptomatic infections after the first dose. While encouraging, there are limitations to this analysis. This is a snapshot of 1 day. It is not clear from the available data whether persons who tested positive without symptoms might have subsequently developed COVID-19 symptoms and therefore have been pre-symptomatic rather than truly asymptomatic. Also, this only shows the effect of the first vaccine dose. Next slide. The grade evidence table for asymptomatic infection is shown here. The certainty of the estimate was graded down 1 point due to serious risk of bias due to selective outcome reporting. Certainty was downgraded a further 2 points due to very serious concern for indirectness. The relative risk of 0.37 favored vaccination. The certainty estimate was type 4. Next. Okay, now we'll move on to grade for harms. Next slide, please. In addition to the randomized controlled trial already described, there were 3 additional studies that provided data on harms. These included an unpublished phase 2 randomized controlled trial and 2 published phase 1 dose escalation open label studies. Next. First, safety data were included from the Moderna phase 3 randomized controlled trial. The safety set included all randomized participants who received at least 1 dose of vaccine or placebo and contributed any solicited adverse reaction data. Analysis was done according to the intervention actually received. Next slide. The phase 2 trial included 200 persons vaccinated with 2 doses of Moderna vaccine and 200 persons who received 2 doses of the placebo. Next. The phase 1 study by Jackson included data on adults aged 18 to 55 years, including 15 who were vaccinated with the relevant dose. And next slide. Finally, the phase 1 study by Anderson included data on adults aged over 55 years, including 20 who were vaccinated with the relevant dose. Next. Okay, this table shows the raw data on the critical outcome serious adverse events. From the phase 1 and phase 2 trials, there were no serious adverse events identified. In the phase 3 trial, there were 147 events among the vaccine group and 153 among the placebo group. Of note, for the grade analysis, we relied on a pooled analysis of the 2 placebo-controlled studies. Next. The grade evidence table for serious adverse events is shown here. There was a balance of serious adverse events reported among vaccinated and placebo, with a relative risk of 0.96 and a 95% confidence interval of 0.77 to 1.20. The certainty assessment was reduced 1 point due to serious concern of indirectness of outcome because the body of evidence does not provide certainty that rare serious adverse events were captured due to the short follow-up and sample size, so the final certainty was type 2. Next. Reactogenicity was evaluated using these same 4 studies. Next. Both of the randomized controlled trials used the same events and grading scales, which are shown here on this slide. The local reactions solicited for the 7 days following vaccination were injection site pain, redness, swelling, and axillary swelling or tenderness, and the systemic events solicited were fever, nausea and vomiting, headache, fatigue, chills, muscle pain, and joint pain. The criteria for defining grade 3 and 4 local and systemic reactions and events are shown on this slide. Next. Grade 3 or 4 local reactions or systemic events were reported in 5% and 6.7% of vaccinated subjects in the phase 1 studies. For the phase 2 study, reactions were reported in 16% of vaccinated and 3% of the placebo group. In the phase 3 study, grade 3 or 4 reactions were reported by 21.6% of vaccinated and 4.4% of the placebo group. Again, grade was conducted using a pooled analysis of the phase 2 and 3 studies. Next. The pooled relative risk for any grade 3 or 4 event was 4.93 with a 95% confidence interval from 4.55 to 5.34. There was no serious concern for risk of bias, inconsistency, indirectness, or imprecision for this outcome. The final certainty was type 1. Next slide, please. This table summarizes our current grade assessment for the Moderna COVID-19 vaccine. In terms of benefits, the available data indicate that the vaccine prevents symptomatic COVID-19 with an evidence type of 1. The vaccine also prevents COVID-19 resulting in hospitalization. For this outcome, certainty was reduced due to indirectness, and the evidence type was 2. For all-cause death, the available evidence slightly favored the intervention, but certainty was reduced for indirectness and imprecision, and the evidence type was 4. No data were available to assess seroconversion. The available data were consistent with a lower incidence of asymptomatic SARS-CoV-2 infection in the vaccinated group, but certainty was reduced for risk of bias and indirectness, and certainty was type 4. In terms of harms, the available data indicate that serious adverse events were balanced between the vaccine and placebo arms, and 3 serious adverse events were judged to be related to vaccination among over 15,000 persons vaccinated. Severe reactions were more common in vaccinated persons, and about 22% of vaccine recipients versus 4% of placebo recipients reported grade 3 or 4 reactions. The evidence type was type 1, indicating high certainty in the estimate. Next, please. To conclude, I would like to put the grade into the context of the policy question, which focuses on an interim recommendation to be issued under an emergency use authorization. Regarding benefits, the phase 3 trial is ongoing, and effect estimates may change with additional follow-up. This raised concern for indirectness of outcome, as ideally we would like to observe efficacy over a longer period than 2 months median. We judged that it is unlikely that the efficacy estimate for symptomatic COVID-19 would change substantially enough in the months following vaccination to fall below the FDA-defined efficacy threshold for an EUA, but indirectness remained a serious concern for some other outcomes. Direct evidence of efficacy for hospitalization and deaths is limited at this time due to the small number of events that has been observed through the cutoff date. From the efficacy against symptomatic disease, we infer that vaccination would also reduce hospitalizations and deaths. Preliminary data were consistent with the vaccine preventing asymptomatic infections. Regarding harms, grade 3 reactions were not uncommon in vaccinated persons. Serious adverse events occurred at a similar frequency in vaccine and placebo groups, but only 3 serious adverse events were associated with vaccination. Next slide. I would like to acknowledge the contributions of everyone listed here, in particular Drs. Curran and Wallace, who have been extremely involved and helpful in reviewing all available data and preparing the grade assessment. Next slide. Thank you.
Thank you very much, Dr. Gargano, for that excellent presentation. I'm going to ask the voting members, do they need any clarification? I don't want to have discussion at this point. We will have that after the presentation by Dr. Oliver. So are there any clarification questions that the work group would like, that the voting group member would like at this point? I see none. Okay, then Dr. Oliver, can we ask you to go forward and present the evidence to recommendations framework for Moderna COVID-19 vaccine, please?
Yes, thank you, Dr. Romero. All right, next slide. Next slide. So I won't go through the details here again, as we've had numerous ETR presentations over the past several weeks. But again, ETR framework provides structure that moves ACIP from evidence to ACIP vaccine recommendations, and will be highlighting updates based on the Moderna COVID-19 vaccine. Next slide. This is the policy question we are addressing today. should vaccination with the Moderna COVID-19 vaccine be recommended for persons 18 years of age and older under an emergency use authorization? Next slide. Again, the PICO question is the focus for the ETR discussion. The population is persons 18 years of age and older with the intervention of the Moderna COVID-19 vaccine, comparison of no vaccine, and the outcomes listed here, which were just detailed in the grade presentation. Next slide. These are the domains for the evidence to recommendation framework that we'll be discussing today. Next slide. And again, we will update to make the questions applicable to the Moderna vaccine and COVID-19 disease. Next slide. So the first domain we will address is the public health problem. Next slide. Addressing the primary question of, is COVID-19 disease of public health importance? Next slide. Every time we've presented this data, the report on overall and daily case counts becomes more somber. As of December 17th, there have been nearly 17 million cases, with the 7-day moving average over 215,000 cases a day. Next slide. I also wanted to show the deaths reported per day. As of December 17th, there have been nearly 310,000 COVID-19 deaths reported to CDC, with the 7-day moving average at 2,600 deaths per day. Next slide. The cumulative hospitalization rate between March and December 12th was 295 per 100,000 population. Among those hospitalized, nearly a third required intensive care and 15% died. The case fatality range is shown here from 0.5 to 1.4%. Next slide. So again, the work group judgment is that yes, COVID-19 disease is of public health importance. Next slide. Next for benefits and harms. Next slide. So in addition to the overall grade presentation, the questions to determine benefits and harm focus on how substantial are the desirable anticipated effects. Next slide. How substantial are the undesirable anticipated effects? Next slide. And finally, do the desirable effects outweigh the undesirable effects? Next slide. So detailed review of the benefits and harms through grade were just presented. We'll walk through highlights and some additional information here. The clinical trial demonstrated high efficacy against the primary endpoint of symptomatic lab-confirmed COVID-19 with an efficacy of 94.1. This was determined to have a high certainty related to the policy question under an EUA. For hospitalization, 10 events occurred, 9 were in the placebo group and 1 in the vaccine group. Vaccine efficacy against hospitalization was estimated at 89%. Given the low numbers and short time of follow-up, there was moderate certainty in this estimate. And for all-cause mortality, deaths were uncommon overall, 6 in the vaccine group and 7 in the placebo group. Given low numbers, there was low certainty in the evidence for this outcome. Next slide. And the ability of the vaccine to prevent asymptomatic SARS-CoV-2 infection has not been assessed to date in a large prospective trial using serial PCRs. However, the ability of the vaccine to prevent asymptomatic infection could be informed by PCR screening among trial participants returning for a second dose. 4 weeks after the first dose of the Moderna COVID-19 vaccine, 14 or 0.1% had a positive PCR without symptoms of COVID, compared to 38 or 0.3% of those receiving placebo. Again, this is one time point prevalence, does not follow people over time, and it's only after the first dose. Protection against asymptomatic infection before or after this time point is unknown. Given all of these caveats, there was low certainty of the evidence. However, it is encouraging, and we look forward to additional data points to inform this outcome. Next slide. For a further summary of benefits, in addition to the efficacy of 94.1 for the primary endpoint, High efficacy was found for additional analyses across age, sex, race, and ethnicity categories, and those with underlying medical conditions. Efficacy among adults 18 to 64 years was 95.6%, and among adults 65 and over was 86%, with wide confidence intervals. To emphasize that these specific estimates are based on small numbers in the trial, and have some variation during the exact population or age cutoff, the efficacy among adults 75 and older was 100%. Most recipients received 2 doses of the Moderna vaccine, but efficacy was estimated at 69.5% between dose 1 and dose 2. Next slide. 30 cases of severe disease were noted in the placebo group and 1 in the vaccine group, yielding a VE estimate of 97%. And the numbers of observed COVID-19 associated hospitalization or deaths were low. However, the trend was consistent, with 9 of 10 hospitalizations and the 1 COVID-associated death occurring in a placebo recipient. Next slide. So next, for a summary of possible harms. Serious adverse events were reported in a similar proportion among recipients of vaccine and placebo. And there was moderate certainty in this evidence. Reactogenicity outcome graded was severe or grade 3 or more reactions. Overall, a grade 3 or higher reaction was reported by 21.5% of those receiving a vaccine and 4.4% of placebo, and this was graded with a high certainty of evidence. Next slide. Local reactions occurring within 7 days were common. Pain at the injection site was the most common. Systemic reactions within 7 days were common as well, with fatigue, headache, and myalgias as the most common. Symptom onset was usually 1 to 2 days post-vaccine receipt, and most symptoms resolved in a median of 2 to 3 days. Next slide. This slide highlights the select local reactions by dose in 2 populations. The top box is persons aged 18 to 64 years, and the lower box is persons 65 years of age and older. Next slide. As you can see highlighted in the red boxes, the proportion of participants who received the Moderna vaccine who reported any local reaction was 75% or over. However, as is highlighted in the blue boxes, 15 to 20% of those receiving placebo reported a local reaction as well. Next slide. This slide shows any systemic reactions and reports of fever in the younger and older populations. Next slide. As shown in the red boxes, around half of vaccine recipients reported any systemic reaction after the first dose, and 70 to 80% reported a systemic reaction after the second dose. However, as again is highlighted by the blue boxes, 30 to 45% of recipients who received the saline placebo also reported a systemic reaction after the first or second dose. Reports of grade 3 or grade 4 fever were rare. And I'll note that as was done after the prior EUA, we will have a website summarizing this reactogenicity data after each dose on the CDC and ACIP websites to help inform providers and patients around possible expected symptoms post-vaccination. Next slide. Lymphadenopathy was also noted in the clinical trial. Ipsilateral, or the same side as the vaccine, axillary swelling and tenderness was a solicited adverse event in this trial, which differs from what was discussed last week. This ipsilateral axillary lymphadenopathy was more common among vaccine recipients less than 65 years of age, occurring among 21% of participants. Grade 3 axillary adenopathy was rare, but slightly more common after the second dose. Adenopathy was noted to last a median of 1 day after the first dose and 2 days after the second dose. As localized lymph nodes are involved in the vaccine response, it is plausible that this could be related to the vaccine. Next slide. Regarding Bell's palsy, there was a small imbalance between the vaccine group, with 3 reports compared to 1 in the placebo group. Currently available information is insufficient to determine a causal relationship with the vaccine. Post-authorization surveillance will be important to help determine any possible causal relationship moving forward. And as has been highlighted previously, serious adverse events were similar between vaccine and placebo group, with no meaningful imbalance between the groups for any specific conditions. Next slide. This slide again highlights the summary of grade from what we just heard, with each evidence type listed. Next slide. So overall, the work group felt that the desirable anticipated effects were large. Next slide. And the undesirable anticipated effects were small. Next slide. Based on this, the work group felt when considering how the desirable effects balance with the undesirable effects, it favors the intervention, the Moderna COVID-19 vaccine. Next slide. Next to the values domain, next slide. Does the target population feel the desirable effects are large relative to the undesirable effects? Next slide. With the second question about important uncertainty or variability in how much people value the main outcomes. Next slide. The details of the methods, which have been presented before, are detailed here. We updated the search through December 18th. Next slide. The proportion intending to receive the vaccine ranged from 40 to 86%, and a recent survey is shown here in the last bullet. 40% reported that they would receive the vaccine as soon as possible, 44% planned to receive the vaccine but wanted to wait a bit, and 15% said they would not receive it. Taking into account people willing to receive the vaccine now and after a brief wait, 84% were willing to take a COVID-19 vaccine. Next slide. This slide combines the data from now 33 surveys with the proportion reporting positive vaccine intentions by month of data collection and bubbles proportional to the survey sample size. The surveys in December report a broad range of vaccine acceptance. Next slide. Many adults report intentions to receive a COVID-19 vaccine with increasing willingness over the past several weeks. Intentions varied substantially by race or ethnicity and socioeconomic status. Next slide. The workgroup interpretation of the first question, does the target population feel the desirable effects are large? The workgroup felt that the answer was probably yes, but likely vary by population and over time. Next slide. So then also felt there was probably important uncertainty or variability in how people value the main outcomes. Next slide. Moving to acceptability. Next slide. The main question for this domain is the Moderna COVID-19 vaccine acceptable to key stakeholders? Next slide. Again, a review of the literature was conducted. Next slide. This information has all been presented previously. There has been no new published information since last week. However, next slide. The media reports over the last week have highlighted numerous healthcare personnel receiving their COVID-19 vaccine. One news report showed that at that hospital, only 2 had declined or deferred vaccination. Acceptability generally focuses on vaccination stakeholders, the persons providing the vaccine. However, in this instance, the stakeholders, healthcare personnel, are also the patients and vaccine recipients. Next slide. And while we had previously been highlighting that jurisdictions had submitted implementation plans, we now report that jurisdictions are implementing vaccination plans. Pharmacy chains are working to launch the COVID-19 vaccine program in long-term care facilities. Next slide. So the workgroup felt that the Moderna COVID-19 vaccine was probably acceptable to key stakeholders. Next slide. Moving to feasibility. Next slide. This domain asks if the Moderna COVID-19 vaccine is feasible to implement. Next slide. The barriers to implementation discussed by the work group included financial barriers, complexity of recommendation, and vaccine storage and handling requirements. Next slide. For financial barriers, as has been discussed earlier, COVID vaccines will be provided free of charge, but health systems or health departments could incur costs for vaccine implementation or clinics. Next slide. Next, for complexity of recommendations, 2 mRNA vaccines under an EUA with different dosing intervals, as well as different storage and handling requirements, may make vaccine recommendations more complex. Next slide. And finally, for vaccine storage and handling requirements, the vaccine must be maintained at standard freezer temperatures for shipping and long-term storage, however, is stable for up to 30 days at refrigerated temperatures. The minimum size of orders is currently at 100 doses. Both of these specifications will have a positive impact on feasibility. However, the requirements of a 2-dose series still remain and could impact feasibility for series completion. Next slide. But overall, the work group felt that the Moderna COVID-19 vaccine was feasible to implement. Next slide. Moving to the resource use domain. Next slide. With the primary question, is the Moderna vaccine a reasonable and efficient allocation of resources? Next slide. There are no new data on the estimated cost associated with COVID-19 disease, but again highlighting here that the estimates remain at hundreds of billions or trillions of dollars. Next slide. And again, the government has already provided billions for the development and manufacturing of vaccines, and vaccine doses purchased with U.S. taxpayer dollars will be given to American people at no cost. Next slide. We will have more information eventually to conduct a full cost-effectiveness analysis. However, again, the work group felt that cost effectiveness is not a primary driver for decision-making during the pandemic. This is an aspect of ETR that will be reevaluated with additional data in future recommendations. Next slide. However, based on the current situation, the work group felt that yes, the Moderna COVID-19 vaccine is a reasonable and efficient allocation of resources. Next slide. And finally, equity. Next slide. The question for this domain is, what would be the impact of the COVID-19 vaccine on health equity? Next slide. Again, through a systematic process and using the EPI of COVID-19, we identified groups that might be disadvantaged in relation to COVID-19 disease burden or receipt of a COVID-19 vaccine. Next slide. The following populations have been identified and discussed in detail at previous ACIP meetings. Next slide. There are specific characteristics of the Moderna COVID-19 vaccine that could impact health equity. First, storage, handling, and administration requirements. This vaccine is refrigerator stable, which will facilitate the ability of the vaccine into more community settings once supply allows. A need for the 2 doses remains a challenge, and follow-up will be challenging for some disadvantaged groups. Next slide. Although COVID vaccines will be provided at no cost, a personal investment in time and travel to obtain the vaccine may be a barrier for some groups. Equity in vaccination program implementation is closely linked. Advancing health equity will require efforts to identify and reduce access-related barriers to vaccination among groups who experience disproportionate COVID-19 related morbidity and mortality. Next slide. The work group felt that the impact of the COVID-19 vaccine would be to probably increase health equity, especially with easier cold chain requirements, allowing for broader availability once supply allows. Next slide. So in summary, next slide. This is a slide of the ETR domains, questions, and the work group judgments from everything that was just presented. Next slide. After reviewing the data, the work group provides a judgment on the balance of consequences. Starting on the left, where the undesirable consequences outweigh desirable consequences, moving to the right, where the desirable consequences outweigh undesirable consequences. Next slide. The work group felt that the desirable consequences clearly outweigh undesirable consequences in most settings. Next slide. In addition, after reviewing the totality of information presented in the evidence to recommendation framework, the work group discussed the type of recommendation proposed to ACIP. The options are: we do not recommend the intervention, we recommend the intervention for individuals based on shared clinical decision making, or we recommend the intervention. Next slide. The work group was in agreement based on the data presented. The type of recommendation proposed is a full recommendation of the intervention. Next slide. Thanks, and happy to take any questions.
Again, Dr. Oliver, thank you very much for another one of your wonderful presentations, clear and lucid. Let me ask the work group to weigh in on any questions or clarifications that they have at this moment. And I see that Dr. Paling has one.
Hi, thank you, Dr. Oliver, for a very clear presentation. I wanted to ask about the vote within the work group. Could you tell us how that came out?
Yes, thanks. So there was when it comes to the balance of consequences, the overwhelming majority felt that the desirable consequences clearly outweigh the undesirable consequences in most settings. There was a very small minority that felt that the desirable consequences probably outweigh the undesirable consequences. And the work group was in agreement based on the type of recommendation.
Thank you. Any other questions or comments? Clarification? Okay. Very good. So, can we have the question that we're going to need to have motions for? You have that ready, Dr. Oliver?
Absolutely. We can pull up the proposed vote slides if that would be helpful.
That would be great. So for the voting members, we need a motion at this point and a second once the question has been read. Let me see here, I'm just making sure. Okay, now, Dr. Cohn, please clarify. At this point then, Doctors Atmar, Fry, and Hunter must recuse themselves from any discussion or vote, correct?
That's correct.
Okay. So, would you read the policy question, please, Dr. Oliver?
Absolutely. So the policy question is, should vaccination with Moderna COVID-19 vaccine be recommended for persons 18 years of age and older under an emergency use authorization? Next slide. As a reminder, these were the work group interpretations. Next slide. So the language for the interim recommendation is the Moderna COVID-19 vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization.
Thank you. So, are there any comments or questions, or does anybody wish to make a motion? And I see Ms. Bata's hand up first. Ms. Bata.
Yes, I would like to move that ACIP recommend the Moderna COVID-19 vaccine for 18 years and older under the FDA EAU.
Thank you, Ms. Bata. Dr. Szilagyi, I see your hand up next.
Yes, I would like to second the motion.
Very good. Thank you, Dr. Szilagyi. So we have a motion and it's been seconded. Is there any further discussion on the part of the voting members of the ACIP? Okay. So then the interim recommendation, the Moderna COVID-19 vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization. Has been motioned and seconded. We will take a vote on this after we have our public comment section. That section is scheduled at half past the hour. Dr. Cohn, we have to adhere to that time, do you not?
Yeah, Dr. Romero, I would like to recommend that we take another 10-minute break right now and resume at 2 p.m., at which time we will do the presentations that are scheduled for after public comment. So we'll have the allocation. Wait, no. Sorry, I'm on the wrong day. The update on COVID-19 vaccines and anaphylaxis and the clinical considerations for use. Then we'll have public comment. And then we can then have discussion around the presentations that are happening at now.
Very good. So for those of you that do not have a final agenda in front of you, so we are going to move the public comment to after the presentations by Dr. Clark and LeMay, and before the vote. So we will break until the hour, and then we'll resume. Thank you again very much.
And to just clarify, the public comment will still occur at 2:30 p.m.
Yes. Very good. Thank you.